Okay, hi everyone. Talking to myself right now, but just in case. Oh, hello. Um, I will be here for the next 45 minutes answering questions. Let's see. So, as a quick reminder, your quiz will only cover digestion, respiration, and circulation. What I mean by that is you don't need to know the stuff before that about animal systems that you'll see on your final exam, but not on this quiz. And then you also don't need to know for this quiz the excretory system, the reproductive system, like all the stuff that we've covered since then. Um, so go ahead and let me know what questions you have, and I'm happy to answer them. Okay. Let me see if I can do this. Hi, hello, Brett. So let me see. Like I said, go ahead and type whatever questions you have. But until someone does, I'll just ramble, I guess, about stuff. So one thing you want to make sure you understand is both the human systems, but also how the systems might work in other organisms. Pretty much focus on your notes. Again, it's, I believe, 132 to 149 in your notes, but nothing else. So nothing before, nothing after. Let's see. Don't forget there are there are practice quizzes, or a practice quiz online that you can take to help you do that. And I will also be online tomorrow, same time, same place, to answer any questions you might have that. Let me see. Okay, so if the mesoderm layer is not present in the animal, is the circulatory system extras? No. So basically what you want to think of is, so in these animals who have these embryonic germ layers, they basically don't have to have mesoderm, but if they don't, that means they're usually a pretty simple animal, like super, super simple, right? So they probably won't even have, excuse me, a circulatory system. Like they were typically animals who only have the 2 layers instead of the 3 will be so simple that they won't have many complex systems. So a jellyfish would be an example where they only had probably 2 embryonic germ layers, so they don't even have a circulatory system. So that's why they don't even have the embryonic tissue to develop that. Gastric juice. So gastric juice is a all-encompassing term to describe the stuff that is produced in the stomach. So you would generally say that gastric juice consists of 3 components. It would consist of your HCl, your pepsinogen, and your mucus. Technically, all 3 of those things are coming from different cells. We didn't go into that on purpose in the lecture. But the idea is just that to sort of cohesively talk about what comes out of the stomach, we just call it gastric juice. So remember that the HCl creates that low pH in order to activate pepsinogen into pepsin. That's going to now allow the pepsin to break down proteins in the food. And then the mucus is produced to essentially protect the stomach, which is muscle and therefore protein itself, from self-digestion, because you wouldn't want the enzymes that are being activated in the stomach to digest the stomach itself. Of course, that does happen, right? That's when you get the ulcers. But that's sort of the idea of it. Okay, what other questions have you got? Go ahead and type them. Let me see. I can think of what else you want to make sure you do. For the remember, there's a really large overlap between the circulatory and the respiratory systems, so you definitely want to be able to sort of talk about how they are connected, as well as individual components of each, right? So I think I mentioned in most classes, hopefully I did, that it's not uncommon for individuals who smoke and therefore having respiratory issues to actually have circulatory issues as well, like cardiovascular issues. Okay, information about the heartbeat. Yes, totally. So heartbeat is, first of all, remember your heart is really unique in the sense that it doesn't have to have anything else to make it beat, right? The heart muscle has the ability to do what we call self-excitation. That being said, then what that means is there is a bundle of cells in the heart called the SA node where your heartbeat originates from. That's why they call it the pacemaker of the heart, because when it fires and sends a signal, then it's going to it's going to cause your heart muscle to contract. So what happens when your heart beats, remember, first of all, is you have the atria on the top and the ventricles on the bottom. So it's essentially two rounds of contraction, right? When the first part contracts and the atria contract, it's going to push all the blood down to the ventricles. And then the ventricles contract, it pushes the blood out to the body and out to the lungs. That being said, so let's go over the heartbeat, right? So it's an electrical signal. That's why they do those EKGs or ECGs, which are electrocardiograms. And so the signal originates in the SA node. The SA node will send an electrical signal, which will flow through the atria to the AV node. When that signal flows through the atria, that is what both the left and atrium at the same time, that's what makes the atria contract, thus pushing the blood from the atrium down to the ventricles, right? From there, it's going to go to the SA node to the AV node to the bundle of His to the Purkinje fibers, and now that's going to cause the ventricles to contract. So if I look at the total pathway of the heartbeat, it's going from the SA node to the AV node to the bundle of His to the Purkinje fibers. And when that whole process happens, is again the atria contract and the ventricle contracts, and now you have basically you're going to have the whole thing, you know, the whole heart now is contracted. How do acoelomates have gastrovascular cavities if they don't have a body cavity? I'm not sure that acoelomates actually have gastrovascular cavities. I could see how some might, but generally I don't think they actually do. I mean, a gastrovascular cavity is the like simplest digestive system you can have, but you know, I think if you don't have a body cavity, you're probably not going to have really the ability to take in any kind of food. So I don't think they actually do. I mean, I'll look that up for you. But when I think of an acoelomate, I think like a flatworm, which is like parasite, which doesn't have an gastrovascular cavity. Animals who have gastrovascular cavities are a little bit more, slightly, more complex. Things like sea anemones or, jellyfish or things like that. Okay, lipase, yes. So essentially, lipase is a lipid digesting enzyme, right? I mean, we're used to the idea of enzymes digesting things. I think with digestion, things just get a little more complicated because now you have bile where it digests the food, the lipids, mechanically, right? So it's taking a big droplet of fat and making a smaller droplet of fat, which we call a micelle. But then in order to really completely digest it, you now need to add in the lipase, which comes from the pancreas. And that is going to, the bile basically creates a larger surface area for the lipase to now digest the lipids into the components, which would be glycerol and fatty acids. So it's sort of like a two-pronged approach, right? First, break the food up into smaller droplets, the lipids, and then now take those smaller droplets of lipids and have the lipase act on it to digest it into its individual components, which would be glycerol and fatty acids, which are then absorbed technically into the lymphatic system first, but then that will join the circulatory system when it kind of dumps into the circulatory system. I think it's right above the heart. Do know your hormones and your digestive enzymes and things like that. Other questions you have? Let's see. Any questions you guys have about like respiration? I haven't seen too many respiration questions yet, so maybe it was kind of steer the conversation in that direction. Is there anything you're not sure about in terms of respiratory stuff? Remember, you do want to, again, as with every system, make sure you understand human respiration, but also respiration in other organisms. So, you know, our lectures are divided up in such a way, right, where we have the non-human stuff and then the human stuff specifically. How does an animal cope with the temperature change in an endotherm? Well, so remember, an endotherm is like us. An endotherm is an organism who needs to maintain a certain internal body temperature. And so in order to do that, they will adjust their, they will basically carry out reactions and processes to keep themselves at that temperature. Whereas an ectotherm, really what it means is they don't have those processes, right? So as long as it's within a certain range, that animal just literally goes with the temperature change. Like it's within a certain range and it's not going to be harmful to the organism. They adjust and just sort of literally go with the flow. Whereas for an endotherm, they would either do things to generate more heat or do things to cool themselves down if the temperature's not right. The bird's respiratory system is kind of confusing. Sure. So remember, the huge thing about the birds is they have these extra air sacs, right? And in having these extra air sacs, what that means is compared to us, like think about how you breathe, right? We take air in, it goes into our lungs, and then we breathe it out through the same pathway. So it's going in and out through the same pathway. Whereas a bird with those extra air sacs now means it's completely changing the way they breathe, because when we inhale, we take air into our lungs. When we exhale, we push it out. But for birds, they have this sort of two-breath cycle, right? So they breathe in the first air, and it actually goes into their posterior air sacs, right? The back air sacs. Then when they exhale, now they're pushing the air through their lungs to their front air sacs, right? Or into their lungs, I should say. And then they inhale again, and they pull the air out of their lungs into their front air sacs. And then they exhale, and they pushes it out again. So what that means then is at any given time, the air that's flowing over a bird's lungs are pretty much like it's fresh air, right? It hasn't been sitting there. It hasn't you don't have any old air that's not getting out because it's all just flowing unidirectionally. And maybe that wouldn't be as big of a deal if you had lots and lots of oxygen all the time. But at the same time, then if you are really high up where there's not a lot of oxygen, and now you need to kind of pull as much oxygen out of the air as you can, you don't really want any air to be old and stale and really kind of just sitting there not doing anything for you. So for a bird, then, we say they have one-way flow through their lungs, and that's really going to allow them to have fresh, like as oxygenated as it can be, air flowing over their lungs at all times. So it's subtle, right? It's not a huge thing, but it's enough that it's going to help them get more oxygen from the air that they're breathing, because they don't have any of the air sort of just sitting there and old and stale. Counter current exchange. Yes. So first of all, it's good to know that while we talk about counter current exchange in the context of the fish's gills and oxygen and things like that, it's a general idea that you'll see in other parts of the body. Sometimes it could also be for heat and things like that. But the overall idea is counter current means that the two things you're looking at, like whatever, in this case, oxygen, for example, is diffusing from and to, is they're going in opposite directions, right? So think of it as like the northbound and the southbound freeway lanes. The reason that this is going to be important for a fish is because, remember, as a respiratory media, water already doesn't have as much oxygen. So it's kind of the same idea as the fish. They have to be able to pull out as much oxygen as they can from this water that doesn't already have a whole lot of oxygen to give. So, well, let's start with concurrent exchange first, right? if you have it in your notes, which unfortunately I can't pull up to show you because I don't think my computer lets me show you what I'm seeing. Basically, with concurrent exchange, the idea would be it's simply the diffusion of oxygen, right, from an area of higher concentration to lower concentration. So if the blood and the water were going in the same direction, then what you see is by about halfway through the gills, the concentration of oxygen in the blood and the water essentially becomes equal. So at that point, you're not going to have any benefit of the gills, like basically, the second half of the gills is not going to do anything because equal amounts of oxygen would move back and forth between the gills and the water. And so that second half of the gills is really wasted. You're not getting any oxygen from the water into the blood. Now, comparatively, if you look at your notes and you look at the countercurrent exchange, right? Even if you just sort of went from water to blood and you drew an arrow from numbers, meaning more oxygen to less oxygen, you'll notice that you can draw an arrow all the way through the gills one from the water into the gills, the whole length. And that's because essentially you can kind of think of it as because the water and the blood are going in opposite directions, the water, like the blood, the water that the blood is seeing, like not really seeing, right? That it's coming into contact with is always like newer water. It's newer in the sense like it hasn't given as much of its oxygen away yet, right? So all the way down the entire length of the gill, you're going to have oxygen being able to diffuse from the water into the gills. And that's really going to maximize, again, the amount of oxygen you get because now you're utilizing the entire length of the gill to get the oxygen because you're always giving it newer water with more oxygen, right? As opposed to if they went in the same direction, they would just level off. So hopefully that kind of clarifies. Oh, and you're welcome. Let's see. What else can I remind you of? Any questions about, well, let's see. Do you guys have any questions about like circulatory stuff? The heart, blood pressure, blood vessels, comparative anatomy, any of that stuff? Sorry, I think I have seasonal allergies. Let's see. What else could we talk about? I think if you look at a lot of the trends across the three systems, you'll notice a lot of times surface area plays a role too. How much detail? I want you to know the diagram in your notes. So I think that in terms of blood glucose level, I want you to know, first of all, so we can actually kind of tie them all together, right? It's the idea that, first of all, you eat something, let's say a carbohydrate-rich meal. You eat it and it gets digested in your mouth, in your small intestine, right? Once you have those component monosaccharides, they are going to be absorbed into the bloodstream by diffusing across into the bloodstream. And so now they've entered the circulatory system, right? That's what raises your blood sugar. Well, two things about that, right? Number one, the sugar is not really doing you any good in your bloodstream, and you want it to go to cells. So at that point, and this is a matter of homeostasis, right? So when your blood sugar levels are too high, then you are going to need to get the sugar into your cells. And the way you do that is you need a key to unlock the cells, and that's insulin, right? So the pancreas would create insulin, and insulin would then serve as like that molecular key that helps the cells of your body now take in the glucose. There's a whole, pathway that does that, but essentially just know that it does that. And in doing so, now you lower your blood sugar levels and you put it back into that range of, the sort of normal range, right? And it's really good at keeping it within a pretty tight range. Well, if now you've, not eaten a while and the sugar is being taken out of your blood for your cells and it's getting lower and lower, Now we would switch gears now, and the pancreas, different cells of the pancreas would relieve glucagon, right? It now does the opposite. So insulin's going to say, hey, let's take the sugar out of the blood into the cells. Let's in the and then it also sends extra glucose to the liver to store as glycogen. Now glucagon's the opposite, right? It's going to say, hey, I don't have enough sugar in my blood. Let me take some out. So it's gonna go to the liver and say, hey, I need to make a withdrawal from the bank, the sugar bank, to take out some of my sugar. So that's what I would want you to know in terms of I want you to know sort of how those hormones play into blood sugar regulation. Systolic and diastolic pressure, yes, I can totally go over that. Remember, ultimately, blood pressure is a measure of how much force is your blood exerting on the walls of your blood vessels, or simply think of it as how much is your blood pushing on your how much is your blood pushing on the walls of your blood vessels, right? And that can be due to a lot of different things. But in the end, think about how much it's pushing on the blood vessel walls. Okay. So that being said, there's actually now two different scenarios for that, right? Because when your heart pushes or pumps, it's going to have a lot more push behind the blood. And so that's going to be a higher pressure because imagine you're squeezing something, right? Right then when you're squeezing, you're applying a lot of pressure. So that would be your systolic pressure. Systolic pressure is measuring when your heart pumps, how much pressure is your blood applying on the walls of your blood vessels. Now, your heart's not constantly contracting, right? It contracts and it relaxes, and it contracts and relaxes. So your diastolic pressure then is essentially a measure of in between contractions, how much pressure is your blood just naturally putting on the walls of your blood vessels. And so those are those two numbers that we get, right? I mean, I don't need you to know the numbers, but the overall idea is that you want to know both are important, right? Because the systolic pressure kind of gives you an idea of how hard is your heart working. Because if your systolic pressure is really high, that means your heart's probably working really hard, pushing with a lot of force. And that would be worrisome because if you have too much, too much force from your blood, it's going to damage your blood vessel, the lining of your blood vessels, and then that can lead to atherosclerosis. And atherosclerosis can then doesn't have to, but can lead to things like heart attacks and strokes. Diastolic pressure is a concern too, though, right? Because if in the meantime, even when your heart's not pumping, there's a lot of pressure on your blood vessel walls, that's also potentially damaging to the blood vessel walls. So that's why doctors are concerned about both systolic and diastolic pressure being too high. Okay, is animal classification? No, it is not. It is in the notes, you're right, but it's not 132 to 149. So again, to clarify, your quiz is only going to be on digestion, respiration, and circulation. Those other things you'll see, but you'll see them on your final exam, part 2. Okay, what else? Sorry. Hormones. I would say know your hormones. We see them show up, a variety of little places, right? There's always, not always, but in many cases, there's hormones involved for each system we talked about, right? Insulin, glucagon, EPO, CCK, gastrin. Yes, I will post the answers to the earlier AP weeks. I think I already have a couple of them up, but I will put the rest up. And then you guys will actually get your AP weeks back soon. I'm trying to get through whatever I can, but obviously at some point I will return them to you because they're more useful to you than me. And we'll also spend the next couple weeks, working on reviewing for the AP exam. You're welcome. Let's see. other stuff. Make sure you can, like I said, I think I mentioned in our video lecture, make sure you know how to read the graphs, like the hemoglobin binding curves and things like that. It's a good question. I think that with animals so it says capillaries' main function is to exchange materials between blood and interstitial fluid. So do animals with open circulatory systems, hemolymph, have capillaries? I don't think they do. I mean, I think that essentially when we talk about like arteries, capillaries, and veins, they're pretty complex tubes, right? And generally speaking, an animal who has an open circulatory system is going to have a much simpler system of tubes for an open system, because it's really just like kind of getting the fluid where it needs to go, but not really, because sometimes it will basically, in doing so, right, it just kind of gets the blood generally to a location, but it's not as directed, so it doesn't have as much of the stuff. So it's probably not going, it wouldn't really have the need for capillaries, right? Because the idea is that the heart pumps, it kind of dumps it out through those tubes, but then it kind of just squishes everywhere. So no, I don't think they have capillaries. Typically when we talk about, the capillaries, arteries, and veins, we are talking more specifically about a closed system. Let's see. What was I talking about before? Oh, you're welcome. You also want to make sure you know how gases are transported, right? So carbon dioxide is transported in the plasma, whereas oxygen, predominantly in the plasma, a little bit is carried by hemoglobin, but for all intents and purposes, it's mostly transported in the plasma, right? So in the case of carbon dioxide, the carbon dioxide is coming from your cells, and then it gets it diffuses in, and then it's going to convert it through a series of reactions, first to carbonic acid, but then carbonic acid becomes bicarbonate, and then the bicarbonate is how it travels through the plasma. So what is the different stuff that happens at the duodenum, jejunum, and ileum, or is it just the same stuff? Typically, the duodenum or the duodenum is where you're going to have the digestion finish up, and then the ileum and the jejunum, I guess, technically is just the absorption, but I don't need you to differentiate between those two locations. I think really I just want you to know that initially you digest it, finish digesting it, and then after that you absorb it. So that's where you'd find like the villi and the microvilli, things like that. You're welcome. Okay. So I guess until people put up another question, I will continue talking about gas exchange. Remember things like partial pressure, right? That it's the same idea as, it's sort of like concentration. I mean, it's not exactly that, right? But I mean, for our general purposes, you can think of that where oxygen and CO2 will diffuse from in areas where you have a higher concentration, a higher partial pressure of oxygen to a lower partial pressure of oxygen. And so oxygen is carried in the in hemoglobin, in the red blood cells, right? On hemoglobin. Each hemoglobin, again, think of it as like a 4-passenger car, where you can it can carry a maximum of 4 oxygens, right? And then and hemoglobin is a very practical molecule where when it has a lot of oxygen around, it will pick it up. But when it doesn't see a lot of oxygen in surroundings, it will drop it off. It's very generous in that sense. So do be sure, again, that you can understand all those aspects, right? Hemoglobin, binding curves, etc. One second, I'm going to wander here for a second and plug in my computer because I don't want us to run out of power halfway through here. Just a second. See if my power cord will stretch. Yep. Okay. Yeah, good. Let's see, what else can I talk about? Let's see. We talked about heartbeat. We talked about digestion, right? Know the different types of digestion, right? Chemical, mechanical, things like that. Know the different blood vessels, how they work, right? The sort of structural components that you would see in each. So, for example, you have like the arteries, right, which have really thick muscular walls because they're going to have this pressure from the heart really pushing on them. So you wouldn't want an artery to burst, and that's why you would, have those really thick muscular walls. Capillaries are going to have the really thin walls because that's where you actually want things to come in and out. And then with veins, you're going to have the one-way valves because in that case, gravity is really not their friend, right? And they really want to make sure that the blood is getting back up to the heart. So they're going to have those valves to kind of help keep it moving along. Let's see. Trypsin. Okay. We didn't go into it too much detail, but essentially trypsin is a protein digesting enzyme. It's in the trypsin is in the, it comes from the pancreas and is deposited into the small intestine, and that's where it's going to help finish off the digestion of proteins. Technically, it's another example. Okay, sorry, I'll answer that question in a second. So technically, it's a good example too, where you have an inactive enzyme that gets converted into an active one, because actually there's a form, it's like pepsin, where there's an inactive form called trypsinogen that gets activated in trypsin. Okay, how is biomechanical digestion? Remember, the idea of mechanical versus chemical is, are you changing it chemically, like into different chemical molecules, or are you just changing the size of it, right? And so technically, bile is a mechanical digestion because all bile does is it takes a big blob of fat and it makes it into smaller blobs of fat, right? So the fat hasn't been chemically changed at that point. It's just a smaller group of it, right? So that's why we call it mechanical digestion. Whereas in contrast, lipase, which comes from your pancreas, is chemically digesting the lipids because it's going to take those lipids and break them down into glycerol and fatty acids. So that's why it would be considered mechanical. Okay, let's see. Lauren, you asked about the AP test. No, I think that, honestly, they used to, right? So the AP exam before 2013 was much more focused on memorizing things and phylums and things like that. They've largely really moved away from that. So I really doubt that they would have that kind of thing. They aren't as interested. They still want to know what you know, but they're much more lenient now in that sense, right? They pretty much want you to be able to read and understand the context of questions. So there's a lot of reading and answering questions about it, as opposed to just having you like spit back facts about different phylums. So no, I don't think they really need you to know all the specific information. I think a lot of cases they want you to have an example in mind for things, right? So but they don't want you to necessarily know all the itsy bitsy details, because a lot of times they'll give that to you in the problem, or they'll give you enough that you can kind of figure out some of those details. I think it also depends on like how old your prep book is. Also, keep in mind those prep books, as nice as they are not written by the College Board, and so they could have essentially errors or things that are not quite correct about them, or they might be over stressing certain things that the AP exam isn't necessarily concerned about. So, in terms of phylum, no, I would say that's not going to be an AP exam, at least not in any sort of really significant way. Yes, blood clotting. Okay. So blood clotting, first of all, is a good example of an enzyme cascade, right? Where like one thing triggers the next, triggers the next. So it's basically, a way to have one thing after another sort of add up, I guess, or like, a chain reaction is the best way to describe it. So the idea is that when you damage your blood vessels, right, you've made a hole in them. That's going to expose collagen fibers, and the collagen fibers, um, what they do is you already have platelets floating in your blood. So now when they pass by that damaged area and they come into contact with the collagen fibers, they get stickier, and that helps them stick, right? So you're going to basically form like a temporary plug where just it's platelets sticking there. But unfortunately, given the way your blood flows, that's not quite enough. So you also need to sort of trigger this whole clotting reaction to fully seal the hole up. And when you do that, then um, what you're actually doing is what you're actually doing in that case is you are creating more of like a mesh net, right? So the platelets are going to release clotting factors, and those clotting factors are now going to basically trigger the prothrombin, which is already floating in your blood, to become thrombin. And then thrombin, which is another hormone or compound, not protein, right, will now act on fibrin or fibrinogen, which is already in your blood. And that converts the fibrinogen into fibrin. So it like forms it into like little threads, right? And that becomes like a mesh net that's going to basically become like a band-aid for the damaged area of a blood vessel. And then platelets and other things will get trapped there, but essentially that's going to plug the hole to stop you from bleeding too much. Because if you bleed too much, you don't have enough blood to pump around. Eventually, though, the area that's plugged up will heal, right? And then that clot is no longer needed. Generally speaking, right, that's a great thing. You want your blood to stop bleeding. You want the whole process to stop bleeding. But keep in mind, remember too, with atherosclerosis, that when you have that high blood pressure and it can damage a plaque, which is that buildup of fatty deposits, now you might accidentally trigger a clotting reaction in like your arteries to your heart or your brain, and that's where you get a heart attack or a stroke. CCK and secretin. No, I wouldn't say that they're examples of the sympathetic and parasympathetic divisions. What you want to think of them instead is they're just hormones that are going to do different things to complete the digestive process, right? So one of them is going to go to the pancreas and say, hey, I need some sodium bicarbonate to neutralize the stomach acid. And then the other one is going to go to the pancreas and say, like, hey, and the liver and say, hey, I need some, digestive enzymes. So, parasympathetic, sympathetic division tends to have to do more with like things like basically like fight or flight, or the opposite, which would be like rest and digest. Okay, we have about 5 minutes left, so any other questions? Did I miss any questions? I think I got them all. Okay, the whole coelomate thing is really, the question is, what does it mean for a coelomate to have a body cavity with mesoderm on both sides? So it's subtle. You're going to look at your picture there, which I don't have. Like I said, I can't show you the picture. But the idea is that when we're getting to pseudocoelomates and coelomates, like you have a body cavity in that case, but is it lined on the inside and the outside with mesoderm, right? And so it's a really technical distinction. In the end, it's just a matter of like, if you only have, mesoderm lining it on one side, remember mesoderm becomes muscle, right? So it just means like with only one layer of muscle, you're not going to be able to move around as well. So that's sort of the significance of it. But also keep in mind the things like coelomate and whatnot are not on the quiz for tomorrow, because that is, I think, not on 132 to 149. It's the part right before that. So again, you'll see that on your final, but you don't need to know for the quiz. Okay, other questions? Let me see if I can think of anything else. Talked about blood vessels. I guess we could talk about other animals' respiratory systems, right? So we already talked about the birds. And then other that, we also talked about insects, right? So insects, don't forget, they have a tracheal system, which means that they have all these holes all over the body, and then they don't use their open circulatory system to transport oxygen, so they can pretty much just like move the oxygen directly to the cells through the tracheal system. Yes, so first of all, if you want to come in anytime during lunch, you can. I think only day I won't be there is Friday. There's like a staff thingy. But other than that, you can come any day during lunch to work on like practice problems and stuff. And then I think what we'll do for after school, for this week, I can be there. Let me double check. Actually, I think I can do that. Let me check my calendar first. But I should be able to be there today on Friday after school if you want to come and do any practice problems there. And then also like next week, I can be there Thursday. No, they're Friday. So again, so every day pretty much, like during lunch, brunch, you're always welcome to do that. And then just something, I guess, further down the line, I haven't mentioned to you guys in class yet, but I will in more detail, is AP review sessions, which aren't really review sessions per se, but like time, essentially. So for me, I will be in the room for about 2 hours on Friday the 10th, and people can just come and ask whatever questions they have, but very kind of informally, and I'll have all the practice materials out there too. And then on Saturday, Mrs. Fallon will do a review session as well, and all students, regardless of who their teacher are invited to both of those. So for Mrs. Fallon's, we're still trying to figure out their location, but she does a little differently, where she has everybody sit together, and she lets you ask questions one at a time, but and she'll start around 2 o'clock, and basically she'll answer questions until all the questions are done. But her policy is that she doesn't answer a question if it's already been asked before. And she said it's usually like a couple hours. Okay, so think of it for FRQs as like you're essentially trying to show them what you know, right? So it's never a bad thing to say more. What I often say is like if you don't write anything, or the less you write, the less they can give you points for, right? So when in doubt, write more. Like standard, you can even see in the paper they give you, like they expect you for any short FRQ for it to be like at least half a page. And then for the longer ones, which they expect you to spend like 22 minutes on, that one I would say you want to write like at least a page and a half, if not 2 pages. But it's also not just about like how long you're writing. you really want to make sure, this is why when you're reading the question, you want to read the question like several times. Make sure you know what they're asking and then like answer that question. Because if you write a really long essay about the wrong topic, they can't give you any points. And I'll show you examples. Like I have examples of what they're looking for. We'll look at some more of the homework ones, but when in doubt, I would say be more in depth, because I think generally students don't do that. But the whole idea is to show them what you know. And so if you're assuming they already know what you know, that's kind of beside the point, right? Okay. No problem. Okay, any last questions before I go for now then? I guess just as a quick reminder, you can come in tomorrow, finish your problem sets. I'll be there lunch, I'll be there brunch. And then tomorrow night, same time, same place, 10:30 to 11:15, I'll be here for any last minute questions you guys have. And then this lecture, I believe, will, upload to YouTube shortly after it's over. So you can also rewatch these if you need to. So, last call. Any questions? You can always email me too. Okay. So if you guys are good for now, then I'm gonna go. No problem. It's late. You all should get some rest soon, huh? But email me if you have any questions. All right. And I'll see you guys tomorrow. Well, I'll see 7th period tomorrow. And 4th and 5th period, of course, come in anytime. All right? Or did I say 4th and 5th? 5th and 6th. All right. Have a good night, guys. Bye-bye.